Nucleus is the most important organelle in the cell, and we find it in almost every cell in your body. The only exception being the mature RBCs. In some cases, like in small lymphocytes or the spermatozoa, the nucleus is so large that it takes up entirely almost all the available space. Now, coming to the structure, the nucleus is protected by a double membrane, that is the nuclear envelope. The inner layer is called as perinuclear membrane, which is having many pores. These pores will allow specific molecules to pass through. And the outer layer or the outer membrane is continuous with the endoplasmic reticulum. Then inside the nucleus, we have DNA. DNA is the chemical blueprint that governs every single function of the cell. Because these DNA molecules are so long, they are packaged with proteins to form chromatin, which then organizes into the chromosomes that we see during the cell division, where the DNA replication, transcription of the RNA, all these processes take place. Then there is a certain darker area inside the nucleus, which is called as nucleolus. What is this nucleolus? So the nucleolus is the site where the RNA processing and the ribosome synthesis is happening. Suppose if the nucleolus is seen prominent and very dark in color under the microscope, that means that cell is producing large amounts of proteins at that time. That is the indication. Then coming to the clinical significance, that means what happens when this nucleus goes out of control, when malfunctioning anything is happening, what happens? That means the disorders of certain DNA can lead to several serious conditions. For example, if the mitosis goes uncontrolled, it can lead to cancer. That's why many anti-cancer drugs are designed specifically to trigger the cell cycle arrest to stop that growth in its tracks. So anti-cancer drugs will target this stage. Then there is the second one is the apoptosis. Apoptosis is also called as programmed cell death. This is actually a natural part of the development and carcinogenesis where the DNA actually fragments to safely shut the cell down. Basically, in natural, if the cell is functioning properly, uh, after its use, after its purpose, the cell will automatically destroy itself. That is the apoptosis. If it is not destroying itself, what happens? It keeps on multiplying. That will ultimately result in the cancers. The third thing is the chromosomal disorders. Chromosomal disorders examples we have trisomies. This happens during the meiosis, where the sister chromatids fail to separate correctly. The separation of the sister chromatids, the process is called as non-disjunction. So a malfunction in the non-disjunction can lead to disorders like trisomy. And then the fourth thing and the most important, the clinical significance of these nucleus is that the mutations. Mutations are basically the errors in the DNA sequence. These changes are the underlying cause of many genetic disorders and also the cancers. The prime example being the sickle cell. So that is in brief about the nucleus. It is a double membrane structure. The inner one is the perinuclear membrane with the pores. The outer one is the continuous with endoplasmic reticulum. Inside there is a nucleolus, which is for primarily for ribosome synthesis, protein synthesis. And then inside the nucleus we have DNA, which is bound to the proteins forming chromatin, which we later will be seeing them as chromosomes. Disorders include mitosis disorders, apoptosis failure, chromosomal disorders, and mutations. Endoplasmic reticulum is actually a network of interconnecting membranes that encloses little channels called as cisternae. Cisternae are the channels that are present in the interconnecting membranes. This network is incredibly vast, running all the way from the outer nuclear envelope right up to the plasma membrane. So we already know that the outer membrane of the nucleus is continuous with the endoplasmic reticulum. So that endoplasmic reticulum will continue until this plasma membrane. So when this endoplasmic reticulum is looked through an electron microscope, it would look like a set of railway tracks weaving through the cytoplasm. As you can see, this is the picture, the rough picture of electron microscopic view railway track appearance. Endoplasmic reticulum is one organelle where, in spite of getting disrupted during the cell fractionation, that means to observe the cell organelles separately, we have to separate them. In order to separate them, we have to break the plasma membrane. That is, we have to break the cell. That is cell fractionation. Then, in this process, sometimes if the cell fractionation is not done properly, there is a chance that the endoplasmic reticulum might break down. And in spite of breaking down, they can reassemble together and can form structures called as microsomes. That is in brief about the endoplasmic reticulum structure. Now coming to the types. We generally classify them into two varieties, that is rough endoplasmic reticulum and smooth endoplasmic reticulum. So first we'll say about the rough endoplasmic reticulum. So rough endoplasmic reticulum, it gets the rough appearance because its cytoplasmic side is studded with many ribosomes. The cytoplasmic side of the rough endoplasmic reticulum is bound to many ribosomes. Then the rough endoplasmic reticulum is the primary site of protein synthesis. Because of this, we will find a very prominent rough endoplasmic. They constantly secrete the proteins, like the plasma cells that keep pumping out the immunoglobulins. Uh, Not just the protein synthesis is its function, it also helps as a construction site for the glycoproteins and the lipoproteins, and it also handles essential post-translational modifications like glycosylations. So the functions of rough endoplasmic reticulum, we can conclude them as site of protein synthesis, secreting cells like immunoglobulins, post-translational modifications. Then we said that rough endoplasmic reticulum is bound to the ribosomes, or ribosomes are bound to the rough endoplasmic reticulum. So what are ribosomes? Ribosomes are basically proteins and ribosomal RNA. In eukaryotes, we have 80S ribosomes. 80S ribosomes, which are made up of two parts, 60S, the larger one, and 40S, the smaller one. The larger subunit is made up of 28S, 5S, and 5.8S rRNA. That is a much parts of the larger subunit, whereas the 40S has 18S rRNA. These are always present wherever the mRNA, tRNA, ribosomes, all these three things are always functioning together because these three are involved in the protein synthesis. Then coming to the smooth endoplasmic reticulum. The smooth endoplasmic reticulum does not have any ribosomes attached. While the rough endoplasmic reticulum focuses on proteins, the smooth endoplasmic reticulum is a powerhouse for lipids. It is a site for synthesizing phospholipids and cholesterol in your adrenal glands and the gonads. That is where the steroid hormones are built. In the liver and the muscles, they manage the glycogen metabolism. In the skeletal muscle, a specialized version called as sarcoplasmic reticulum stores and releases the calcium, which is needed for every move we make, that is the muscle contraction. And it also handles both fatty acid elongation and also the desaturation. Then finally, the last function is it acts as the cell's primary detox center, especially in the liver and the kidneys. And how is it doing that? It uses an enzyme system called as cytochrome P450. Cytochrome P450 to hydroxylate and neutralize drugs like barbiturates and phenytoin. Now, coming to the clinical importance, clinical significance, here there is the cytochrome P450 can be induced by alcohol. That means more cytochrome P450 will be activated, more and more drugs are neutralized, and that will make the treatment ineffective. So that's why the endoplasmic reticulum function is very important as it is directly impacting the medical treatments also. Apparatus was first described way back in 1898 by Camillo Golgi, a discovery that earned him the Nobel Prize in 1906. The Golgi can be described as a network of flattened smooth membranes and the vesicles. It acts as a converging area for the endoplasmic reticulum. If endoplasmic reticulum is the production unit, then the Golgi complex acts as the primary packaging unit. So production is happening in the endoplasmic reticulum, whereas packaging is happening in the Golgi apparatus. So how is this process started? The process begins as the nascent proteins move through the endoplasmic reticulum. Here the proteins are synthesized. From endoplasmic reticulum, they start their journey towards the Golgi complex. During this journey, some carbohydrate groups are added to them, turning them into glycoproteins, that is, proteins plus carbohydrates. These glycoproteins are then transported into the Golgi apparatus for storage. Once inside the Golgi apparatus, the newly synthesized proteins are sorted, they are separated based on specific sorting signals encoded within them. That is, after being sorted, they are packaged into vesicles, each equipped with different types of coating proteins, packed into coated vesicles, each equipped with different types of coating proteins to ensure they get where they need to go. All this transport is an energy dependent process. There are four paths the transport can be occurring. The first is they can be directly secreted to the surrounding medium, direct secretion. They can pass through the cell membrane directly. That is the first path. Then the second one is they can become part of the cell membrane. One is they can go to the cell membrane. The second one is they can become part of the cell membrane. That means acting as a structural support, or they can be packaged as vesicles. First, they can be packaged as vesicles, and depending on the stimuli of secretion, they will be secreted, broken down and secreted. The best example is the insulin secretion. Then finally, the last thing is they can be collected into lysosome packets. That means cellular digestion. Whenever something is not needed or whenever the function is finished, they can act as waste disposal systems also. Then coming to the clinical significance, there is something called as eye cell disease. The eye cell disease, also called as inclusion cell disease or mucolipidosis 2 disease. This is a disorder of Golgi function caused by failure to deliver the enzymes to the lysosomes. That is, instead of reaching the lysosomes where these enzymes belong, the enzymes are accidentally secreted outside the cell. Because of this shipping error, the condition is characterized by two major issues. That is, a severe deficiency of the lysosomal enzymes inside the cell and accumulation of the glycosaminoglycans and glycolipids in the connective tissues like fibroblasts, which can lead to several health complications. The mitochondria are small spherical rod-like bodies and usually 0.5 micrometers in diameter. They are not present in every cell. So which are the cells where they are absent is mature RBCs. The mature RBCs actually destroy their mitochondria through a process called as mitophagy to make room for oxygen transport. On the other hand, the tail of the sperm cell is absolutely packed with them to provide the energy needed for the movement. The mitochondria is called as the powerhouse of the cell. Why? Because they take the energy from the food we eat and trap it as a chemical energy in the form of ATP. To do this, they use a unique double membrane structure. The outer membrane acts as the skin. The inner membrane folds into structures called as cristae, which contains the enzymes needed for the electron transport chain. Then inside the fluid matrix contains the materials needed for the citric acid cycle or urea cycle or sometimes heme synthesis also. That is the matrix, outer membrane, inner membrane, and the matrix. Mitochondria are truly unique because they have their own specific DNA and their own ribosomes, which are different from the ones in the rest of the cell, that is in the nucleus. While your cellular DNA controls most things, the mitochondrial DNA handles the division of the mitochondria themselves. Because their protein synthesis is so similar to bacteria, certain antibiotics can actually inhibit the mitochondrial protein production without affecting the rest of your cell. And finally, a mitochondrial DNA is passed on entirely only from the mother. That is by a process called as maternal inheritance. So what happens when the mitochondrial function is impaired? It can lead to many serious clinical issues like inherited disorders. Mutations can cause conditions like fumarase deficiency or Lyon disease, affecting how the body processes the energy. Then again, apoptosis. Mitochondrial dysfunction is actually one of the earliest signs that a cell is undergoing apoptosis. Then poisons. Some of the deadliest poisons like cyanide and the carbon monoxide, they work by inhibiting the cytochrome oxidase, thereby decreasing the ATP. Then in cases like viral hepatitis or alcoholic liver disease, we can see mega mitochondria. That means mitochondria are larger than their usual size, but they have an impaired function. They are non-functional mitochondria, hence they are of no use. And finally, since RBCs lack mitochondria entirely, they have to rely only on the anaerobic glycolysis for all their energy needs. Peroxisomes are tiny spherical organelles between zero point 0.3 to 1.5 micrometers in diameter. While they are small, they are extremely active, especially in cells like leukocytes, platelets, where they act as the front line for the chemical detoxification. Within our body, peroxidation of polyunsaturated fatty acids can lead to the formation of hydroperoxides. Hydroperoxides, free radicals. These free radicals are dangerous as they can damage our cell membranes, tissues, and even our genes. So, to prevent this, the peroxisomes are packed with the two specific enzymes, that is catalase and peroxidase. These enzymes hunt down and destroy the unwanted peroxides or free radicals before they can cause permanent damage. Hence, one of their biggest roles is free radical scavenging. Then they are also useful for many metabolic pathways. The first one being oxidation of long chain fatty acids. Oxidation of long chain fatty acids, long chain, 26 carbon chain length or 28 carbon chain length. So long chain fatty acids oxidation and even degradation of the amino acids. So oxidation is one of the functions. Then hydrogen peroxide metabolism. The catalase decomposes the H2O2 to water, harmless water, and oxygen immediately. Then the peroxisomes also help in the biosynthesis of bile acids and plasmalogens, which are the critical components for the healthy cell membranes. Then we have 3 and 4. Then we have disease prevention. By maintaining these balances, the peroxisomes play a major role in preventing the genesis of the cancers and various neurodegenerative disorders. And the membrane function, there are certain there are certain peroxisomal proteins which have certain membrane functions and organelle interaction. So that is also one of the functions. Then what happens if all these functions get disorganized? When these organelles malfunction, it leads to severe metabolic disorders known as peroxisomal biogenesis disorders. Out of which the first one is adrenoleukodystrophy. We'll discuss first about adrenoleukodystrophy. Adrenoleukodystrophy is a rare condition where deficiency of the peroxisomal matrix proteins leads to buildup of very long chain fatty acids. Very long chain fatty acids get accumulated, and deficiency of certain proteins and enzymes will cause insufficient oxidation. This will cause progressive degeneration of the liver, kidney, and brain. Then the second one is Zellweger syndrome. Zellweger syndrome is also called a cerebrohepatorenal syndrome. This is a severe inborn error of metabolism where the proteins fail to be transported into the peroxisomes. This leaves the peroxisomes empty. The patients typically show neurologic hepatic dysfunction and most do not survive beyond 1 year of age. Here also there will be accumulation of long chain, very long chain fatty acids. Then the last one is primary hyperoxaluria. Primary hyperoxaluria occurs due to defect in how the peroxisome metabolizes the glyoxylate, which is derived from the glycine, amino acid glycine. So from protecting our DNA to synthesizing vital bile acids, peroxisomes are the unsung heroes of the cellular health. Understanding their function helps us to see exactly how our bodies manage the toxic threats at a microscopic level. The lysosomes are essentially the bags of enzymes that act as the cell's personal incineration units, breaking down the unwanted molecules and the cellular debris to keep the environment clean. Most of these enzymes are acid hydrolysis and they require a very specific environment to work, usually an acidic medium, that is pH of approximately 5. The acidic enzymes basically include polysaccharide hydrolyzing enzymes like alpha glucosidase and lysozyme. That is, those are the polysaccharide hydrolyzing enzymes. Then we have protein hydrolyzers such as cathepsins and collagenase. Then nucleic acid hydrolyzing enzymes like ribonuclease. Then lipid hydrolyzers like the phospholipases. So once again, polysaccharide hydrolyzers enzymes are alpha glucosidase and lysozymes. Protein things are cathepsins, collagenase. Nucleic acids are ribonuclease. Then lipid hydrolyzing are phospholipases. As long as the lysosomal membranes stay intact, these powerful enzymes they stay encapsulated. But if the membrane ruptures, the enzymes leak out and can begin digesting the cell's own tissue, leading to significant damage. So how do they work? So how do they work? Oh, just a second, I'll check that. How do they work? When a cell takes in a foreign particle, it forms an endocytic vesicle or phagosome. This then fuses with a primary lysosome. This then fuses with a primary lysosome to create a secondary lysosome. This is also known as digestive vacuole. Inside the particle is progressively broken down until the cell can utilize the fully hydrolyzed products. So when the things go wrong, it can lead to several diseases. First, for example, let's take about the gout. So butyrate crystals deposit in the joints, and when the cells try to dissolve them, the crystals physically shred the lysosomal membrane. This releases those acidic enzymes, causing the painful inflammation and arthritis associated with the disease. Then second one, we have tumor metastasis. Lysosome also plays a role in tumor metastasis. Certain cancer cells, they liberate proteases called cathepsins. Once outside, these enzymes degrade the basal lamina by eating through the collagen and elastin, literally clearing a path for the tumor cells to travel through to distant parts of the body. Then we have genetic and acquired disorders, genetic conditions like eye cell disease. Here the enzymes synthesized correctly, but they lack a specific marker. Golgi unable to target the enzymes due to the mannose-6-phosphate deficiency. Basically, this mannose-6-phosphate deficiency acts as a marker or uh, acts as a marker to tell where the enzyme should reach. When this is absent, these enzymes they remain in the blood and can reach anywhere else. That's why these are important. Then we have certain lysosomal storage disorders like Gaucher's disease or Pompe's disease, where a missing enzyme leads to toxic buildup of lipids or polysaccharides. These get accumulated, and sometimes even environmental factors also play a role. That is silicosis. Inhaling silica particles causes the lysosomal rupture in the lungs, leading to fibrosis, lung fibrosis, and loss of lung elasticity. And finally, autophagy, the cells controlled recycling program where the lysosomes play the lead role. There are four main types to know. There is a microautophagy where the lysosome directly takes up the cytoplasmic proteins. Lysosome directly takes up cytoplasmic proteins. That is the microautophagy. Then chaperone mediated autophagy, a selective process for specifically flagged proteins. Then macroautophagy, when an autophagosome is formed to deliver large amounts of unwanted material for breakdown. Large amounts breakdown. That is macroautophagy. Then finally we have mitophagy, the selective housekeeping of the damaged mitochondria. Without this mitophagy, the toxic protein aggregates would build up, contributing to conditions like cancer, Alzheimer's, and other age-related disorders.